List 4 types of biological molecules.
There's carbohydrates, there's lipids, there's proteins, and there's amino acids.
For each of the biological molecules, identify the general name of the monomer and the name of the bond that holds the monomers together.
For carbohydrates, the monomer is monosaccharides, and the bond is glycosidic linkage.
True.
For lipids, the monomer is fatty acids, and the bond is ester linkage. For amino acid, so for proteins, the monomer is amino acids, and the bond is peptide bonds.
Yep.
And for nucleic acids, the monomer is nucleotides, and the bond is phosphodiester bond or
Nice.
linkage.
Yeah, you got those. Okay, differences between hydrolysis and condensation or dehydration synthesis reactions.
Hydrolysis is the addition of water to break bonds, and that usually breaks polymers into monomers. And condensation is usually the combination of two monomers to form a polymer by removing water.
Yes.
It forms bonds.
Yep. Forms removes water. Yes. Okay. Identify the storage and structural polysaccharides.
So storage saccharides are there is there is starch and glycogen.
Yep.
And for structural is cellulose and chitin.
Yep. Okay, determine the type of glucose isomer, alpha or beta, found in starch, amylase or
Amylopectin, yeah.
Yes. Glycogen and cellulose, as well as the type of alpha or beta linkages found.
Okay, so in starch and in glycogen, there is an alpha 1,4 linkage, but for branching amylopectin and glycogen, there's a 1,6 alpha linkage, and then for storage like cellulose and chitin, it's beta 1,4 glycosidic linkage.
True. Yep, that is all right. Okay, tell me the structural differences between unsaturated and saturated fats.
Okay, for saturated fats, there is no double bonds, and it's more solid at room temperature. For unsaturated fats, there's one or more double bonds, and it's more liquid at room temperature.
Yep, that's good. Define amphipathic and provide examples.
Amphipathic is when a substance has both hydrophobic and hydrophilic elements in it, and an example would be steroids.
Yep, that's good. Okay, general structure of an amino acid.
An amino acid has an amino group, has a carboxyl group, and has an R side chain group, which depicts its polarity.
Yep. What are the primary, secondary, tertiary, and quaternary stages of protein folding?
So primary is basically a linear line of polypeptides. Secondary is when they're bonded together by nitrogen and hydrogen bonds. Tertiary is when they're all folded together to form like a sort of 3D shape. And the bonds are from the intermolecular forces between the amino acids, and also they have hydrogen and hydrogen bonds, and like the reactions between the different amino acids. And quaternary is basically subunits of the tertiary structure combined together, where the bonds from different polypeptides link together to form a quaternary structure.
Nice. Okay, what are the basic features found in all cells?
Basic features. Every cell has ribosomes, DNA,
Yep.
plasma membrane.
Yep.
Hold on, I know this. There's DNA, ribosomes, plasma membrane, and cytoplasm. Cytosol.
Yeah, you got it. Okay, what's the difference in characteristics between prokaryote and eukaryote cells?
Prokaryotes they have no membrane-bound organelles. The DNA, they don't have a nucleus, so the DNA is like this sort of singular strand of chromatin that's located in the nucleoid region. And for eukaryotic cells, they have membrane-bound organelles, and the DNA is packaged like linear chromosomes, which are made of chromatin, and that's located in the nucleus.
Sweet. And that is all. Good job.
Yeah.